In Chapter 2, we're going to discuss the actual mechanism by which DNA, single-stranded, double-stranded now together, double-stranded DNA, is going to replicate itself. And in order to replicate itself, the first thing it's going to have to do, that double-stranded DNA, is, of course, to separate. Okay? So in this particular section, we're going to discuss the various enzymes and processes involved in doing this. And then we're also going to discuss how the DNA, which is pretty good, the DNA polymerase is pretty good, but it's not perfect. We're going to discuss how mechanisms that repair the DNA operate. So if you compare DNA replication between prokaryotes and eukaryotes, you see that they're fairly similar. There's similar mechanisms of replication. For example, they're both semi-conservative. What does semi-conservative mean? Well, what it means is that when you take a strand and you duplicate it, each of the new daughter strands will have one old piece of DNA and one of the newer pieces of DNA that was synthesized. So replication is semi-conservative. One of the old strands of DNA is conserved in each daughter cell. Replication in both prokaryotes and eukaryotes is also bidirectional. That means that if you have a replication fork opening, it's going to be advancing in both directions. So the replication fork is a dynamic structure, but it's going to be going in both directions for both prokaryotes and eukaryotes. So that's where the similarities sort of come to a close. The overall process of replication is very similar. You're always going to be tested about the differences, though. So let's point out some key differences. In prokaryotes, you see that the DNA is circular in structure. It's circular. Whereas in eukaryotes, it's linear. As a matter of fact, you know that in eukaryotes, we sort of divide our DNA into chromosomes. So each of the chromosomes is a linear structure. They're not a circular structure of DNA. They're linear structures of DNA. Another key difference is the origins of replication. In prokaryotes, there's a single origin of replication, of course, going bidirectionally, where the replication fork opens, whereas in eukaryotes, there's multiple origins of replication. Sometimes these origins of replication are called replication bubbles, because when you actually look at them under the electron microscope, under high enough magnification, they look like little bubbles of DNA that are opening. Because the eukaryotic genomes are so large, you need multiple origins of replication, otherwise you never get the complete genome duplicated. So, bacteria, single origin replication, eukaryotes, multiple origins of replication. And of course, at the end of replication, you see two circular genomes from the prokaryote, whereas at the end of replication in eukaryotes, what you end up with are sister chromatids. You'll review this when you look at medical genetics, but sister chromatids are when a double-stranded DNA, a single double-stranded DNA molecule, duplicates itself. Okay, at the end of that duplication step, you end up with two sister chromatids. They should be identical to each other, and they are joined together at a centromere. It's these sister chromatids that should be separated with the mitotic spindle during mitosis. So this is what should occur in a typical cell. One other little piece of information I want to add here is the structures that are at the ends of the chromosomes. What do we call these? Well, we call the sequence of DNA at the ends of the chromosomes telomeres. Telomeres are basically sequences of DNA at the ends of the chromosomes, and these act sort of as a buffer. The telomere sequence doesn't really encode any protein that we know of, but the telomere sequence instead acts as a buffer. So as the cell ages, the telomeres are going to get shortened. And actually the length of the telomere is sort of like a timekeeper that tells us how old the cell is. Cells that have DNA with very short telomeres are older cells. So what you'd associate telomere shortening with is the age of a cell. As the telomeres shorten, the cells are aging, and they eventually are going to undergo apoptosis. Now, a key way that they could ask you about this is to ask you about the activity of an enzyme called telomerase. Telomerase is an enzyme that actually, it sounds like it should break down telomeres, but no, it actually makes telomeres. So telomerase is the enzyme that makes the telomeres. There's only two cell types that have telomerase activity associated with them that you have to worry about. Number one is germ cells. So in germ cells, you don't want the telomeres to shorten because these are germ cells that are going to eventually make a new organism. So there's telomerase activity to lengthen the telomeres in the germ cells. The other cell type where you see telomerase activity, much more clinically relevant, is within tumor cells. Tumor cells are going to have telomerase activity oftentimes stimulated. Why is this problematic? Well, if you picture a healthy cell, over time is going to accumulate mutations. But over time, the healthy cell is going to have shortening of their telomeres. So the cell's accumulating mutations, the telomeres are shortening, and eventually the cell's going to call it quits, and it's going to undergo apoptosis. What happens if in a tumor cell, though, the telomerase activity gets stimulated? Well, now the cell's accumulating mutations, the telomeres are shortening, but the telomerase activity is stimulated. So the telomeres are going to be lengthened. Well, now, instead of the cell getting short telomeres and undergoing apoptosis, the cell's going to be tricked into thinking that it's perfectly healthy and that it's a young cell. But the problem is it has mutations. And some of these mutations are going to be oncogenic. So that's an association that you should be able to make. Telomerase activity is associated with oncogenesis of cells. Once again, let's look at our cell cycle. And this is looking at eukaryotic replication during the S phase. So during the G1 phase of the cell cycle, remember we had a cell with double-stranded DNA. We're going to have a short arm and a long arm to each chromosome. And the cell may or may not enter into the G0 phase and just kind of hang out and do its thing. Okay, now, some sort of signal is going to come along, a growth factor, a hormone, and it's going to stimulate the double-stranded DNA to duplicate itself through mechanisms we'll talk about shortly. During the S phase, you're going to end up with two sister chromatids. These are each double-stranded DNA entities. They're joined together by a centromere, and they should be identical to each other, assuming there's been no mutations during the DNA synthesis phase. Of course, the cell's going to progress through G2, and eventually during mitosis, these are going to be pulled apart. These sister chromatids are going to be pulled apart via the mitotic spindle apparatus, and each daughter cell is going to end up with one of these sister chromatids. Remember, the cell's starting out diploid because why? Because even though there's shown just one chromosome, there's also going to be a homologous copy of each of the chromosomes. One that was inherited from the maternal and one that's not shown here that was inherited from the paternal side. So the cell is diploid at this particular stage, and it remains diploid throughout the entire cell cycle. Contrast that from meiosis. In meiosis, remember, the cells will, at the end of the cycle, will become haploid. Once again, an electron micrograph here. This is showing you, although it's difficult to visualize, this is double-stranded DNA that's already been duplicated. So shown here is the picture, and shown here is the cartoon which depicts this duplicated chromosome. So this chromosome is separated, is joined together. Each of the sister chromatids are joined together at a centromere, and these are eventually going to be pulled apart during mitosis. If you look at the substructure of a chromosome that's been duplicated, you'll notice that there's a P arm and a Q arm to each chromosome. One of these is a long and one of these is a short arm. Can you remember which is long and which is short? Well, the term P actually was derived from petite. Okay, the French word petite meaning small. So the P or the petite arm is actually the small arm of each chromosome. Whereas the Q arm, it just happens to be the next letter in the alphabet, the Q arm is the long chromosomal arm. So keep that in mind.